Hello everyone and welcome back to my channel and a new video. So today's video is about Krebs cycle or also called as TCA cycle, that is tricarboxylic acid cycle. So let's start. So this is the Krebs cycle. Before starting the actual Krebs cycle, we will first see about a process called acetylation which is performed just before Krebs cycle. So in acetylation, pyruvic acid which comes from glycolysis pathway enters and gets converted to acetyl coenzyme A, this compound. Through the process or reaction called dehydrogenation, in the presence of enzyme pyruvate dehydrogenase. And in this step, NAD plus gets converted to NADH2. So this process is dehydrogenation, conversion of pyruvic acid into acetyl coenzyme A in the presence of pyruvate dehydrogenase. This process is called acetylation. This process is also carried out in mitochondrial matrix along with this actual Krebs cycle, which we will see now. So, this acetyl coenzyme A fuses with the first member of Krebs cycle called oxaloacetic acid, that is OAA. So this is the first member of Krebs cycle. This gets condensed into second member which is citric acid. So the reaction is condensation, that is addition of H2O with oxaloacetic acid in the presence of enzyme citrate synthase. This is the first step. Second is conversion of citric acid into cis-aconitic acid in the presence of enzyme aconitase and catalyst Fe2+. Now, in this step, dehydration takes place, that is removal of H2O, one molecule of water. This is the second step. Third step is conversion of cis-aconitic acid into isocitric acid. This is the third compound. Here, hydration takes place, that is again addition of H2O. This is the first hydration in the TCA cycle in the presence of enzyme aconitase again and catalyst Fe2+. Now fourth is conversion of isocitric acid into oxalosuccinic acid. Now here dehydrogenation takes place. This is the first dehydrogenation, that is conversion of NAD plus into NADH2 in the presence of enzyme isocitrate dehydrogenase and catalyst Mn2+. Now fifth step is conversion of oxalosuccinic acid into alpha ketoglutaric acid. Here decarboxylation takes place, that is removal of one molecule of CO2. This is the first decarboxylation in this cycle in the presence of enzyme oxalosuccinate decarboxylase, because decarboxylation is taking place. Sixth step is conversion of alpha ketoglutaric acid into succinyl coenzyme A. CoA means coenzyme A in the presence of enzyme alpha ketoglutarate dehydrogenase. So here both decarboxylation and dehydrogenation takes place. Decarboxylation means removal of one molecule of CO2 and dehydrogenation is conversion of NAD into NADH2. So this is second decarboxylation and second dehydrogenation. Seventh step is conversion of succinyl coenzyme A into succinic acid. This is the seventh step in the presence of enzyme succinyl thiokinase. Now here This coenzyme A gets removed from succinyl coenzyme A and only succinic acid remains. And here GDP gets converted to GTP. Now GTP is nothing but ATP. So 1 GTP is equal to 1 ATP. And this is an important step because this is called as substrate level phosphorylation. Which step? Seventh step of Krebs cycle. Eighth step is conversion of succinic acid into fumaric acid in the presence of enzyme succinate dehydrogenase. So here again dehydrogenation takes place. This is the third dehydrogenation of Krebs cycle. Here FAD plus gets converted to FADH2. This is the eighth step. Ninth step is conversion of fumaric acid into malic acid in the presence of enzyme fumarase, and here hydration takes place. This is the second hydration in the cycle. Hydration means addition of one molecule of H2. This was the ninth step, and the last 10th step is conversion of malic acid into again OAA, that is oxaloacetic acid, and then the cycle continues. So this step is done in the presence of malate dehydrogenase, and here dehydrogenation takes place again. So this is the fourth dehydrogenation in the Krebs cycle. Here NAD plus gets converted to NADH2. And then this Krebs cycle again continues. So this was Krebs cycle, and Krebs cycle is also carried out in mitochondrial matrix. Glycolysis takes place in cytoplasm, and Krebs cycle and acetylation takes place in mitochondrial matrix. Now here are some important points to remember which can be asked as questions in NEET exam. So first, in acetylation, 2 NADH2 are produced. So we know 1 NADH2 gives 3 ATPs. So 2 NADH2 will give 6 ATPs. So the net gain of ATPs in acetylation is 6 ATPs. Now TCA cycle, that is Krebs cycle. Here 6 NADH2s are produced, so total 18 ATPs. 2 FADH2s are produced, so 4 ATPs, and 2 ATPs, that is GTPs, are produced. So total 18 plus 4 plus 2. So total net gain of ATP in TCA cycle is 24 ATPs. Now you may have a doubt that in the Krebs cycle we saw only 4 times dehydrogenation take place, that is 3 times NADH2 was produced and 1 times FADH2 was produced and 1 times GTP was produced. But the pyruvic acid which came, so this which comes from glycolysis is 2 molecules of pyruvic acid. So from glycolysis 2 molecules of pyruvic acid enter into acetylation and Krebs cycle process. So therefore whatever TCA cycle, whatever compounds are there, they will also be produced in 2 molecules. So therefore 3 NADH2 into 2, 6 NADH2s, 1 FADH2 was produced into 2 FADH2s, and 1 GTP was produced, so into 2 GTPs. So in total 24 ATPs is the net gain. Now remember, this can also be asked, how many dehydrogenations we saw? So 4. 3 of NADH2s and 1 of FADH2. And we saw 2 decarboxylations in the Krebs cycle. So these were some important points to remember in Krebs cycle or TCA cycle. So if you like this video, click on the like button and don't forget to subscribe my channel to get notifications of more such useful new videos. Thank you.